Hello, biology students! Now we're going to talk about active and passive transport in more depth. Let's learn the difference. So we mentioned briefly last time that active and passive transport are the main types of transport that can go through the cell membrane. However, let's learn about them more in depth. The first type, passive, does not use ATP. It moves along the concentration gradient from high to low. And the examples that we've been learning, diffusion, osmosis, and facilitated diffusion with the help of a transport protein, all move from high to low concentration. And since that's the normal direction, they use no ATP. Imagine this is like being on a conveyor belt or a lazy river going the regular normal direction of the stream. High, where there's more, to low, where there's less, is the natural movement and does not require any energy. Active means that we have lots of movement, we need energy. This is going the opposite direction. Instead of going down the slide from a high position to a low position, we're going up the slide from where there's less to where there's more. This is really hard, all right? This is called against the concentration gradient because it's going the opposite direction. It's like moving against the direction of a river or a stream or against a conveyor belt or an escalator. It would be hard, doable but hard, and it takes energy. Some of the examples we're going to be learning about here are gated channels and endo and exocytosis, which we'll learn about in the next couple of slides. But in general, we're always going from where there's less, look at these two blocks that are orange blocks, there's less, to where there's more of the orange blocks, which is the opposite direction that they naturally go, so it requires energy. Let's look at some examples. The first type, and remember, they all go against the gradient from low to high, and all require energy, is protein-gated channels. This one is just the movement through a protein gate. So just like facilitated diffusion used a channel protein, this one will too, but it's moving the opposite direction. So notice that these orange ones, there's less of them, and these green ones, there's less of them on this side, and they're moving both of them with the help of ATP to the side where there was already a lot. So now there's even more green things over here and even more orange things over here. So same thing as facilitated diffusion, but now I'm using ATP and I'm going the opposite direction. So I call it a protein gated channel because it's a gate that needs ATP to open the lock, like a key in a lock. The next type, endocytosis. This is how large particles enter a cell. This has the phrase endo, which sounds like the word enter. So this is how a cell might eat something, such as an amoeba or other types of single-celled organisms. They eat something. Enter. Endocytosis. Last one, exocytosis, is how large particles exit the cell. Exocytosis sounds like the word exo, exit. This is how we get rid of waste quickly out of the cell that are big waste particles. Let's check for understanding. Is this endocytosis or exocytosis we're seeing based on the direction of the arrow movement? Oh, it's exiting, so it must be exocytosis. Exit, exocytosis. Is this exo or endocytosis? Oh, it's endocytosis because the arrow's pointing in. Enter. All right, well, hopefully you're getting it. We will practice more in class. Great job, guys.